You've probably heard that some people have a really hard time digesting milk. And even worse, that means they probably will have a hard time digesting perhaps the best food of all, ice cream. What's at the heart of this issue? Why can't some people digest milk like other people can? Well, as we'll find out, it's all about the enzymes. Let's get started. So in the last video, you may remember we talked about catalysts. Catalysts are used to reduce activation energy to help a reaction occur. Now in biology and biological systems, the catalysts that we often refer to are called enzymes. So in this graph, you can see what's happening. An enzyme essentially lowers the activation energy required for a reactant to turn into some products. So in this case, you see some reactants, right? Those eventually are going to turn into some products. But to get this reaction to take place, an enzyme, as you see here, helps provide the space so that that reactant can break down and we get some products. Notice that the enzyme in this case is still available for use in the next reaction. So in this video, we're really going to focus on everything we need to know about enzymes. So what's the basic function? How do they work? So as you can see here, enzymes are really just these biological catalysts made of protein and a little RNA. That's what the enzyme is composed of. The thing that binds to the enzyme is called the substrate. After it's bound to the enzyme, we call that the enzyme substrate complex, and the reaction occurs. The enzyme, notice, is not degraded, but our product has changed. So these enzymes essentially are facilitating the reactions in the body by lowering that activation energy. And what you'll find is that most of our reactions in the body will require some sort of enzyme. Now, what we also know is that the enzymes are highly specific. Notice the space is almost like it's been made to fit the particular substrate. So enzymes are highly specific. Only certain substrates will fit in certain enzymes. And of course, you've got thousands of different enzymes in your cell. The other nice thing about enzyme is that the body makes it, and then it can reuse it over and over and over again. So even though there is a little investment, right, a little energy needed to make these enzymes, they're reusable, so we get a lot of bang for our buck with enzymes. So how does a substrate fit in? How does that work? So a substrate is basically just the reactant that binds to the enzyme, all right? So it's just the fancy word for whatever chemical, whatever reactant is binding to the enzyme. And then it forms that temporary enzyme substrate complex. So let me give you a very specific example. Okay, here's our enzyme. And notice we have a place here where the substrate, in this case sucrose, is going to bind. We call that site, the place where the substrate binds, we call the active site. So that active site is where sucrose is going to come and bind. So here we go. We've got sucrose, which you know is just a disaccharide. Sucrose joins. Notice it joins to the active site. And now what happens is, if you look very closely, the enzyme provides the place for a reaction to occur. In this case, it's a hydrolysis reaction. We're just breaking that bond between the two monosaccharides that make up sucrose. And you know that we'll leave here with glucose and fructose. So the reaction was sucrose needs to be broken down into glucose and fructose. What helped it? The enzyme helped it. Now, another thing that we might consider is that many of our enzymes will end in ASE. That's a way that we can generally identify something's an enzyme. So this enzyme that you see here is sucrase because it breaks down sucrose. We'll see lots of enzymes as we move throughout biology, and many of those will end in ASE. So back to my very beginning slide about the ice cream. Why can some people digest milk? Some people can't. Well, there is a particular enzyme called lactase, which basically breaks down the milk sugar lactose. So some people's DNA actually doesn't code to make a lot of lactase, the enzyme that breaks down lactose. So those people, unfortunately, when they have a lot of milk sugar or lactose, won't be able to break that down. It sort of sits in the gut and the stomach. And since your body can't break it down, well, some other opportunists, right, some bacteria in the gut might start breaking that down, putting off a lot of gas as a byproduct, and those people that are lactose intolerant will feel pretty bloated and pretty uncomfortable because they can't break that down. Now, you might have also heard that there is something that people that are lactose intolerant can do. They can take Lactaid, which is just a pill that has the enzyme lactase. So you might be able to have a little milk if you take the enzyme by mouth and break it down naturally in the body. Okay? So how does this work? Well, we actually call this an induced fit model because the enzyme kind of changes its conformation a little bit, the 3D structure, to fit the substrate. So here, let's say we've got a substrate, and notice, and remember, everything in the body is really surrounded by lots of water. So there's our substrate, it's got a lot of water around it. Here's our enzyme. Now it looks like that's going to fit right in, right? But the enzyme's got lots of water. So what happens is, as the substrate moves over, you're going to see a conformational change. And the enzyme will actually stabilize and bond with hydrogen bonds, also some ionic and hydrophobic interactions, to make this substrate fit very well so the reaction can take place. We call this an induced fit model because really the enzyme's kind of like a glove that's fitting right around that substrate. So lots of things affect how enzymes work, right? Enzymes being mostly protein and a little RNA. Well, we know proteins have shape, they can be malformed, they can change. They're most effective at certain conditions. So these are all the factors that affect enzymes. Well, if we want more reaction to take place, one thing we can do is actually increase or change the enzyme concentration. More enzymes means more reactions. That makes sense. Another thing we could do is change the substrate concentration. So in our previous example, the substrate was sucrose that we were breaking down. So if we want more reaction, we can increase the amount of sucrose, right? So if you want more reaction, increase the enzyme, increase the substrate. Also, the temperature affects the rate at which enzymes take place. Now I'm going to go over each of these a little individually in a moment, but I wanted to give you the general overview. pH, right? How acidic or basic a solution is will affect how the enzymes work. How salty, right? The salinity. And then two others, activators and inhibitors. Activators, such as things like vitamins, can actually activate enzymes. Inhibitors, some poisons and toxins, can actually inhibit the enzyme from doing its work. So what I want to do in the next few minutes is actually go through each of these and explain how they work. But these 7 factors affect enzyme functioning. So, enzyme concentration, it just makes sense. If the enzyme concentration goes up, the reaction rate goes up. Makes sense. You get more enzymes, you're going to get more reaction. And this is a typical graph of what that would look like. Now notice that it generally kind of is going to level off. And that's normally because we've either run out of substrate to react or all the enzyme active sites, right, are full. Okay? Or the substrate concentration has run out too. So the other thing we can do is increase the substrate concentration. These graphs look very similar. As the substrate, in the previous case, the sucrose, as that went up, well, we're going to get more reaction, right? More breaking down of that sucrose. And eventually, this will probably level off too. Again, and that's probably because either the enzyme active sites are full or we've run out of substrate, either one. So increase the enzyme, increase the substrate to get more reaction to take place. Temperature is also important. Why? Because enzymes function best at certain temperatures. Enzymes evolved to function at temperatures in the organisms where they are. So if you take most of the human enzymes, they function best between 35 and 40 degrees Celsius, right, which is our body temperature, 37 Celsius. So they sort of have an optimum temperature. Can they work a little lower? Sure. Can they work a little higher? Sure. But notice there's an optimum temperature where you're going to get the greatest reaction. If we increase too far beyond the ideal temperature, that heat will denature the enzyme, right? And it will lose its shape. So denaturation is a fancy word for the folding of the protein. The 3D shape is going to be denatured, and it's going to be unfolded or not folded correctly. So if we get too high of a temperature, it's going to be denatured. If the temperature is too low, remember back to chemistry, molecules are moving slower when we have a decreased temperature, so they're going to collide fewer times, not going to get as many collisions, therefore not get as many reactions, right? So enzymes have an ideal or optimum temperature, which will give us the greatest reaction. Enzymes also have an ideal pH. So if you think about pH, how acidic or basic something is, the pH changes the charges of these enzymes, right? Remember pH, the hydrogen ion concentration. This can disrupt bonds, change the 3D shape, and affect the tertiary structure. So if you think about it like a general enzyme in our body, it's probably going to work about a neutral pH, right? Think about enzymes in our blood. We have a pretty neutral pH of our blood. Those enzymes are going to be optimal about a neutral pH of 7. However, if you think about it, there are places in the body where the pH is very low or a little higher. Think about, for example, the stomach. Pepsin is an enzyme in the stomach that breaks down protein. Well, you've probably heard a lot about stomach acid, right? The stomach is very acidic. So this enzyme evolved to actually function at a very low pH in the 2 to 3 range. We also have another one, trypsin, which is in the small intestine. And this enzyme also breaks down protein. But notice it functions at a slightly more basic pH. Its optimum temperature is 8 because that is, its optimum pH is 8 because that is the pH in the areas of the small intestine where trypsin works. So pH can also affect the enzyme ability to do work. Now, activators, the name pretty much says what it does. It activates the enzyme by creating a change in the structure of the enzyme. There are 2 types of activators I really want you to know. The first one is a cofactor. A cofactor is an inorganic, right? Think no carbon, inorganic. An inorganic compound or a non-protein or an ion that binds in the enzyme to increase the reaction of the enzyme. And a prime example is iron in hemoglobin. Iron, you see close to these heme groups in the hemoglobin. That helps the hemoglobin do its job, which is, of course, to carry oxygen in the blood cells. So without iron, right, you're not going to have as effective hemoglobin molecules at carrying the oxygen. Another one that we see a lot is magnesium in chlorophyll, which you know is in plant cells, right, to photosynthesis. So cofactors help enzymes function by binding to certain places. Now you also have coenzymes. That might be a little hard to see. Sorry, I wrote over it. Coenzymes, I'll rewrite it. Coenzyme with the enzyme, right? So these are organic molecules that bind to or near the active site, to the enzyme near the active site that help the enzyme function. So when you think of people taking vitamins, vitamins are actually coenzymes that help reactions take place. So there's NAD, vitamin B3, and when we learn a little bit more about the metabolism in the next few chapters, we'll find that NAD is also an electron carrier that is involved in metabolism. Okay, so the activators, two types. Cofactors are small little ions or inorganic compounds. Coenzymes are larger organic molecules like vitamins, which bind near the active site of the enzyme to increase the rate. So we have activators to activate enzymes. Well, then the other thing must be, of course, inhibitors. They do what the name suggests as well. These are regulatory molecules that will affect the enzyme activity. So normal binding, if we look, the substrate, of course, binds to the enzyme in the active site, active. Well, there are two types of inhibition that I want to talk to you about. Competitive inhibition means some other molecule, is competing in the active site. So notice that this enzyme is inactive because there's an inhibitor, there's a competitive inhibitor competing for the active site, so the substrate can't bind. That's competitive. Non-competitive means some inhibitor has bound somewhere else, and we call that an allosteric site. And it changes the shape of the enzyme so that the substrate no longer fits correctly. This is an inactive enzyme. So let me go over these two, competitive and non-competitive inhibition, a little deeper. So competitive, remember, is when the inhibitor and substrate are competing for an active site. Prime example of this is penicillin, right? The drug you take that's an antibiotic to kill bacteria, because bacteria use a certain enzyme to build cell walls. Penicillin is a competitive inhibitor for that enzyme, so it blocks it. So this is the normal function, right? Enzyme plus the substrate, then we get a product. But if you have, as you see, the competitive inhibitor, this substrate can no longer bind. They are competing for the active site. This inhibitor, right, that's the inhibitor right there, inhibits the substrate from binding. It's competitive. A non-competitive inhibitor binds somewhere other than the active site. We call that place the allosteric site. A lot of poisons, toxins, and some of our new anti-cancer drugs actually are allosteric inhibitors or non-competitive inhibitors. It causes the enzyme to change its shape. So once again, here's the normal thing happening, right? An allosteric inhibitor or a non-competitive inhibitor will bind to the active site either before the substrate has bound or after, and it changes the shape so that the substrate doesn't fit or work anymore. So this can bind before the substrate even comes in, or in this case, notice that the substrate has already been bound, already trying to react. Then the inhibitor can come on and say, hey, all right, I know you're reacting, but we don't need this anymore. Go ahead and inhibit this from taking place. We don't need this reaction to take place. So this is non-competitive because it is not competing for the active site. So to wrap things up, let's think about how these chemical reactions can take place. Enzymes are never generally just sort of a one-stop shop. There are often many enzymes put together to create some sort of pathway. So generally, your body will do a lot of chemical reactions organized in a pathway in a small step. So let's say we had this one molecule that the body needed to ultimately convert to this, right? So maybe it's an amino acid that we need to turn into another amino acid. Well, that will take place through several small steps, right? This turns into this, which turns into this, which turns into this, which ultimately gives us this. Well, believe it or not, there are enzymes along each step to do the work. So enzyme 1 will convert molecule A to molecule B. As you can see, enzyme 2 converts B to C. What does that look like? Well, if you look, all right, here is molecule A that we want to convert. Notice it would fit right in the active site of enzyme 1. Once that's happened, it converts it to molecule B. Okay, molecule B will then bind to the active site of enzyme 2. Enzyme 2 will convert it to this, which will then bind to this enzyme, which converts it to this, which binds to this enzyme, and ultimately we end up with molecule E. So notice that all these enzymes are close together, this case in a membrane, and it just is a series of steps that converts one molecule to another. Why is this important? Because it's efficient. They're all close together when we need to take this to this, right? But it also gives us regulation. And the way it gives us regulation is here in our last slide. It allows for feedback inhibition. So for example, let's say we're converting this amino acid here to this amino acid here. Well, if we're doing this conversion, we will ultimately get to the point where the number of this amino acid is very high. Like, we don't need any more. We've got a lot of this. We don't need to make any more. So what will happen often is that the abundance of this molecule in the pathway, the end molecule, will actually give a signal or give some feedback that we don't need to do this anymore, and it will act as an inhibitor. So what you'll see here is that that molecule will go over, bind to that enzyme, change the shape so that this no longer will bind. So in essence, it's turning off this entire coordinated production because I don't need to go through all these steps anymore to make E if I've got enough of this molecule E. This is called feedback inhibition, and it's actually one of the ways enzymes are used to regulate and coordinate production of certain molecules in the body. Okay, so hopefully this video helped you understand the basics of enzymes, all the factors that affect enzymes, and then how enzymes can be inhibited by non-competitive and competitive inhibition.